All right, guys, let's do this. So DNA replication is exactly what it sounds like. We are replicating DNA. So DNA replication is the process of DNA making more DNA. Why does DNA even need to make more DNA? No, really, I'm asking you. Why does DNA need to make more DNA? Think about it. Hopefully because you remembered that sometimes a cell needs to duplicate. A cell needs to make another cell. And when it needs to make another cell, it's going to make a second copy of the DNA so that it can pass that on to the daughter cells. Okay? So the next question then is, when does that happen? So when specifically, meaning in the cell cycle, when is that going to happen? Yeah, legit, I'm asking you another question. Okay, so you probably went interphase. Hopefully you went fancy pants and you said during the S phase of interphase. So remember we said the whole function of the S phase of interphase is this is when the DNA replicates. So I sort of alluded to it before and made it sound all magical. Now is how you're going to find out how that happens. Okay, so during the S phase of interphase is when the cell makes more DNA, and that's because the cell is getting ready to replicate and make a new cell. All right, so the first thing that needs to happen is that our DNA helix needs to unzip. So the helix is all unzipped there, and it's not going to unzip entirely. Some of it will unzip. The rest of it is going to stay together, because if you can imagine, you already know how crazy and chaotic DNA is when it's in that chromatin form. But then it's going to split apart. If it splits apart, now you've got twice as much crazy and chaos. We don't need that. So we're just going to split a little bit at a time. Then there's an enzyme called DNA polymerase. DNA polymerase. So you know what a polymer is. A polymer is many pieces put together. ACE tells you you've got an enzyme. So it's an enzyme that puts things together. And then what's it putting together? It's putting together DNA. So DNA polymerase. So what DNA polymerase does is it comes in and it's going to attach individual nucleotides to the strands that are hanging free. So DNA polymerase is an enzyme that comes in and goes, hey, what are you? Here's a friend, and attaches it. Hey, what are you? Here's a friend, and attaches. What are you? Here's a friend, and attaches. So it's just going to pull nucleotides and attach them onto these strands as is appropriate with those base pairing rules. So remember that with those base pairing rules, adenine bonds to thymine and cytosine bonds to guanine. So here we have a DNA polymerase working on this strand, and it saw this adenine and so bonded a thymine there. It saw this cytosine and bonded a guanine there. Saw this thymine, bonded an adenine. Then you have another DNA polymerase up over on this other strand, saw a cytosine, so it attached to guanine. Saw an adenine, so it attached to thymine. Saw a guanine, so it attached to cytosine, and so on. So this one's attaching and this one's attaching. Now you may have noticed that that's happening in two different directions at the same time. Because remember, those two strands run opposite to each other. So our template strands, our original strands, go opposite to each other. So this template strand is going this way. This template strand is going this way. That means that the addition on this one needs to go this way, and the addition on this one needs to go this way, to keep everybody going the opposite direction of whoever they're bonded to. Your new strands are called complementary strands. So see, your originals are the templates, your new ones are complementary. Replication is complete when you've got two daughter strands left. So just like you had daughter cells when you did the cell cycle and you had cell replication, you've got daughter cells, now you've got daughter strands. So the daughter strands are now, we're talking about a new double helix. So the daughter strand is not the same as the complementary strand. The complementary strand is attached to the template strand, and those two together make the daughter strand. So each daughter strand consists of one original template strand and one new complementary strand. Okay, so we call this process semi-conservative, because conservative would mean we're saving everything and the template stay together, where we would be conserving everything exactly the way it was. With semi-conservative, we've got one old and one new, and they're attached to each other. And then we've got another old and another new, and they're attached to each other. That's better than having old and new, because if you had old and you did that over and over and over again, that old and old would start to get broken down, and you'd have a lot more mutations going on, and you'd be making copies of all those mutations. If we do it this way, we've always got somebody fresh and new in there, and so we're always keeping things good, and we've got less chance of mutations that are going to be harmful. So, quick overview, you've got your original DNA, your two strands are running in the opposite direction to each other. That DNA is going to unzip. Notice again, our strands are still running opposite to each other, they've just unzipped. You've got complementary base pairs getting added in two different directions because they're opposite of the two strands. And then when you're finished, you've got two new daughter strands. So again, you have a template and a complementary strand that make up a daughter strand. And then here you've got a complementary strand and a template strand that makes up the daughter strand. And really, that is all there is to DNA replication as far as you're concerned.